But we've got a couple of tasks left here in chapter 10, and then hopefully we'll have some time to transition into chapter 11 at the end of today's lecture. So chapter 10, we're looking at toxic materials again, and we've got just a few left. And again, we're in the camp here of chronic materials, latent materials, things that develop over time. And one of those things is asbestos. Now, asbestos itself is not a singular compound or even a single set of atoms. It's a mineral. It's a rock. It's found in a lot of different materials. Basically contains magnesium, silicon, oxygen, and a couple of other things. But the ratios of magnesium, silicon, and oxygen can vary from sample to sample. So it doesn't really have a true chemical formula per se. It just, it is what it is. Now, the problem with asbestos, now asbestos is primarily used for its insulating capabilities. Asbestos itself is a pretty good insulator. In fact, we have some asbestos samples upstairs, and you'll actually see them in action when you do your oxidizers lab. But we have some asbestos samples upstairs, and they're heat resistant enough that you can basically have them on fire. They really don't catch on fire. The demonstration that you'll see in the lab is we'll actually do fireworks. We'll make some homemade fireworks out of the raw materials and some chemical know-how. And we'll do the reaction right on top of the asbestos board. Won't notice a thing. So as a building material, it was used primarily because of this heat-resistant kind of power. Problem with it is that it does degrade over time. And when it degrades, it forms these little tiny fibers that are small, they are buoyant, they float in water, and we generally call these forms friable fibers. Friable or friable fibers have a lot of severe health hazards associated with them. One of the main reasons that they are is because of their buoyancy. They're able to travel in your blood. They're able to stay in your respiratory system. And what's nasty about them is they're just the right size to get stuck in your bronchioles, to get stuck in those air pockets and scar up your tissues and leave you susceptible to other kinds of attack. So you can see that on the scale of microns, these friable these friable fibers are roughly the same size as a bacteria would be. So we're talking about that kind of small. Not quite as small as your finer sands, but pretty small nonetheless. And on the agency, actually I take that back, I'm reading the scale wrong. Fine sand is actually bigger. So asbestos, we can't see it, just like we can't see bacteria. Those little fibers that get made are smaller than what our eyes can see. Now, once they get into your lungs, they can start damaging your tissue systems. And again, this is where we get things like lung cancer. We can also get other illnesses, things like asbestosis, which is just the scarring of the lung tissue, because those fibers get inside of your lungs, get inside of those little bronchioles, and just kind of start cutting up the tissue. In addition to lung cancer, we can also see mesothelioma, the favorite of your pick your big time law firm on TV. Throw a stone, you'll probably find 3 mesothelioma commercials in the span it takes to have that stone hit the ground. Now, mesothelioma is a form of cancer, that affects primarily the chest and the abdomen and the membranes of those organs. And it can be nasty stuff. And certainly if you have a family member that's gone through that, it's hard to watch. Because of its link to cancer, asbestos has been banned in most places as a building material, as any other kind of material use. When asbestos is present in the workplace, OSHA requires that we post it. If you have asbestos in your home, which a lot of people do, especially if their houses were made and are older than, about mid-century, 1980s or so is when they stopped really using asbestos as a primary building material. My house was built in the 1960s. When we bought it, it had asbestos tile flooring. And when you have asbestos in your home, you don't necessarily need to remove it. On its own, it doesn't pose a whole lot of threat to you unless it starts to degrade and disintegrate. When it does, then it does pose a problem to you. So if you undergo a renovation in your house and they find asbestos, then they normally need to bring in somebody who has an EPA approval to remove it safely from the house. But in some places, it's just best to kind of leave it alone rather than tear it up or try to remove it yourself, or try to remove it, period. If it's just sitting there, in our case, we just covered up that tile with carpeting. And that carpet and the carpet pad provide the barrier between us and the asbestos. We don't have to worry about any of those side effects from asbestos when we're in our basement. If we had tried to rip the floor up ourselves and replace it with peel and stick tile or something like that, then yeah, we'd have a much bigger problem on our hands. Because that whole removal process is going to create dust. It's going to create fine particles, and if you breathe that stuff in, you're going to breathe in asbestos, and there are risks involved with that. For the last section here in chapter 10, we're going to switch gears. Everything we've talked about to this point have been naturally occurring products, things that you would encounter either in your daily life or in your workplace environment or something along those lines. These last couple of examples here, however, represent biological warfare agents, things that are created either artificially in a laboratory or occur naturally in some kind of way and are derived from living things. These kinds of agents, etiological agents, that's how you pronounce that word, can cause disease or can produce toxins that will ultimately injure incapacitate, or cause mass casualties. Scary thing about a lot of these biological agents is that because they are biological, they need to work through your body's systems. It may take you a while to recognize it, anybody to recognize that they are there. So you may not actually know that you were exposed to it until days later you start to develop symptoms. What are some common examples of these? Well, before we get to that, how would these be distributed? Yes. Okay. That would be method number 3. You inoculate fabric, paper, or other items with the infectious material. So Josh is thinking of anthrax. Probably the most famous anthrax scare here in the United States occurred in 2001, 2002, where anthrax-laden envelopes were sent to a number of members of our government and our news media. Oh, go ahead. Possibly. I could be confusing with something else. I thought that was cyanide. Okay, that's a little bit different. That's not a biological agent. That's a different kind of toxic methodology. Other ways, aerosolization. If you saw the first Christian Bale Batman movie, the Batman Begins, the biological agent that Scarecrow used was basically an aerosolized neurotoxin that made everybody hallucinate. Also fits into the second one, the contamination of food and drinking water. Batman villains are really good at this. So there are lots of ways that we can get these kinds of agents out there. And because they're biological, they have a means of surviving kind of on a longer scale than others. All they need is to be exposed to a host. Once the host absorbs them, inhales them, however, they can go to work on that person and start to degrade their immune system. When infectious substances are transported, they have to carry the biohazard placard as seen here. This is the most common pictogram for indicating that we have a biological agent that is being moved. When these kinds of infectious substances are transported, they are usually transported in a 2-fold or 3-fold packaging system to help prevent their spread in the case of an accidental kind of rupture. So you can see that you've got a primary receptacle here. This is some kind of absorbent packing material, something that is going to be able to keep moisture out. Could be something like styrofoam, could be something like a soft absorbing kind of cardboard, could be a number of different things. Inside of that contains the waterproof culture specimen tubing. That goes inside of a secondary package. The secondary package tells what is in it, where it came from, who sent it. That is usually made out of some kind of a rigid cardboard of some sort, something that is also relatively water resistant. And then finally, all of that is packed into a larger third box that is usually of some kind of rigid material, potentially encased in packing material, styrofoam, something that would prevent accidental tipping and shock and that kind of thing. So all those precautions are in place so that in the event that this gets bumped or dislodged or otherwise runs into some kind of catastrophe, maybe it gets flooded, the chances of the infectious substance getting out, or you opening up all the packaging and finding a vial that has been broken and is open, and now all that infectious substance is just out in the air, out in the packing material, chances of all that are pretty low under the triple packaging system. All right, let's talk about anthrax. Anthrax is a bacterial substance, Bacillus Anthracis. It can be absorbed through your skin. You can ingest it. You can inhale the spores that come off of anthrax. So all three modes of entry are possible with anthrax. Obviously, as is the case for most substances, the inhalation hazard is the most dangerous. Inhalation anthrax is the most dangerous and the most lethal form of the disease. Bacteria are getting directly into your bloodstream, and they can go after your body systems much more directly than if they have to be digested or absorbed through the skin first. Another common form of biological agent is something called ricin. Ricin is not a bacteria itself. It is a biological agent, however. It is derived from a particular variety of the castor plant, Ricinum Communis. So what they do is they can actually extract the toxin from the castor plant, and then where it becomes particularly dangerous is that can then be converted into a mist or a powder or a pellet. It dissolves in liquid. It can be injected into muscle. And as a toxin, there isn't a specific antidote for it. Yes, sir. Yeah, I can't say I'm not surprised by that. Um, you know, I don't know what other uses the castor plant in this case would have other than something like castor oil, but they're um, ricin was in the news media. It's been a few years, but there was a relatively short ricin scare. And the thing with any of these terrorist plot scares is just about knowing how widespread they ultimately are. In the case of anthrax in 0102, this is coming off immediately off of the heels of the terrorist attacks on 9/11. And then this happens. It looks like it's a coordinated effort where a number of people are being targeted. And it turns out it wasn't as coordinated and targeted as, say, the 9/11 hijackings were. Ricin, same kind of thing. We had a couple of people that were given, I think it was, I want to say it was envelopes as well for ricin. I can't remember exact details. But a couple of people in a very short period of time were exposed to ricin through the same kind of form of media, and the same kind of thing happened. News caught on to it. People were put on alert. Turned out to be a pretty isolated incident. It was one person. Wasn't a massively coordinated effort. So that more or less concludes for us chapter 10. There are other kinds of toxins out there. There are other kinds of biological and toxicological agents that we could talk about at greater length. But these are probably the most common ones. I chose anthrax and ricin because, in particular, here in the United States, we've dealt with scares of both of those on a large national scale. And even though 9/11 was now 20 years ago, it's still relatively fresh in the public memory. Most of you, if I say anthrax, have at least some idea of where the origin of that story came from. But we're going to close the book on chapter 10 right there. Before we move on, any questions about toxins, toxic materials, or other things that we discussed here in chapter 10?